Hello, everyone. I'm Steve Anderson. I'm the Chief Scientific Officer for Covance, the Drug Development Division of LabCorp. And I'm glad to be here today to talk with you about the continuum that we see in the drug development space and diagnostic space from the discovery of biomarkers all the way through the development of companion diagnostics, which really gives us, that continuum gives us a roadmap for the delivery on precision medicine. What I'll be covering today is indicated here on my first slide. I'll provide a little bit of background on some of the key trends in precision medicine. This whole idea of discovery of a biomarker, qualifying it, moving it through the development pathway, and where appropriately, developing it as a companion diagnostic. I'll talk about the current landscape in companion diagnostics. And I picked two case studies around trends in biomarkers and companion diagnostics. One focused on work that's being done today in immuno-oncology with checkpoint inhibitors and other immune modulating therapies. And then work that is also ongoing but offers great promise in precision and personalized medicine, which are the applications in cell and gene therapy. I'll then pivot and talk a little bit about those factors that really are relevant for the utilization and uptake of lab tests, biomarker tests, and companion diagnostics to help in clinical patient management and how those are commercialized. And then I'll finish up with a few thoughts on future directions. Sorry about that. So some drivers in the area of precision medicine. If we stay at the top part of this slide, we now know that there are over 300 biomarker tests that are in current drug labels. These biomarker tests or assays have a variety of uses ranging from helping to discern the likelihood of an adverse event, to monitoring patients on a therapy, to stratifying patients with regard to prognosis or likelihood of response, to those that are used to select patients for a safe and effective use of a new therapy, such as a companion diagnostic. On the top panel, we see that there's over 40 companion diagnostic assays already in use today, and that number continues to grow. And in the middle, we see an area that is continuing to expand, the delivery of novel therapeutics that are targeted at alleviating genetically defined diseases like rare disease, and in oncology, adoptive cell therapies directed at key features of a tumor in order to eliminate those tumor cells from that patient. In the lower right-hand side, we see that biomarkers are really a critical portion of the drug development process, and it's really driven by a variety of reasons. One is the understanding of biomarkers. Those biomarkers help us understand and define disease biology. They are also linked to potential new therapeutic approaches. And lastly, when a therapy comes to market in a crowded space, it can be a differentiator of that new asset, that new therapy in the marketplace. In the lower left-hand side, we see that the sort of march towards the increased use of precision or personalized medicine approaches in drug development. In 2018, it crossed the threshold, so that of the 40% threshold, so that of those new molecular entities that received FDA clearance, now over 40% of them had a personalized medicine approach. In 2019, it was 45%, and that march to an increased level of precision and personalized medicine continues to march forward. So we think about biomarker discovery all the way through commercialization of a diagnostic assay that's relevant for drug development. So I've broken this into three phases. It's very simplistic, but exploratory, clinical development, and commercialization. In the exploratory sense, we're discovering biomarkers. The translational scientists are determining their use. They're leveraging those assays to determine mechanism of action, proof of concept, the appropriate platform to assess that biomarker as it moves into the clinical. In the clinical space, we want a robust assay that can be applied operationally in a global trial setting so that we can select and identify and stratify the appropriate patients for enrollment in those trials. It's also relevant to consider other factors like logistics, platform, etc., so you can bring these assays to a broad patient population. And then lastly, as we think about this, as a new therapy comes to market with an associated biomarker assay, how do we commercialize that appropriately? How do we transition from that clinical trial space into routine diagnostic testing? And what are those factors that are relevant for a commercial success? And I'll finish up with some thoughts on that, as we come to the end of this presentation. So in the area of precision medicine, there are a variety of things that are driving our thinking around biomarkers. On the left-hand side, these are more related to the translational sciences and clinical development programs. And then on the right-hand side, it really is more about the technologies that are being applied. So we know that there are new therapeutic approaches, whether they be targeted therapies. We see an increased number of new targeted therapies coming to market, whether they're immune modulating therapies, whether they're adoptive cell therapies, and novel approaches to engage both the immune cells and the tumor cells. We know that we're starting to be focused on combination therapies, looking at how those combination therapies increase likelihood of success or increased efficacy while minimizing toxicity. And then lastly, we know that whether it's a basket trial, an umbrella trial, an adaptive trial, biomarker tests are being incorporated into and are driving complexity of trial execution, really as we think about things like tumor agnostic, biomarker driven assays. So it's not the site of the disease or the site of the cancer that's driving the thinking, but the biomarker data. And then on the right-hand side, we look at how new technologies are being applied. We see more effort on next-generation sequencing or multi-analyte assessment, so that as we have more and more biomarkers that are relevant for stratification and patient selection, if you look in non-small cell lung cancer with the number of targeted therapies and immunotherapies that are available, the ability to assess that entire landscape in a single broad spectrum assay is relevant for better patient management and enrollment in trials. In addition to NGS, next generation sequencing, we have other tools that allow us to look at proteomic-based assays where, again, we measure multiple analytes in a single assessment, which provides a more accurate depiction of the biology. And then liquid biopsies or other technologies that provide ease of use for patients and for execution of drug development trials also are becoming more and more relevant today. I'm going to focus a lot in the area of oncology, but we see the same principles now being deployed and driven into other therapeutic areas. That being driven by our better understanding of those other therapeutic areas or disease biologies, whether it be immunology, cardiovascular disease, rare diseases. But in oncology, for how we think of incorporating biomarker science into drug development. And I love this slide. I've shown this in many presentations. We've seen a rapid evolution over the last several years in how trials in oncology are designed and executed. So if you look on the left-hand panel, the traditional chemotherapy or cytotoxic trials had a standard dose escalation and expansion paradigm from first in human to registrational trials. The phases for the drug development were, again, phase 1, first in human, phase 2, and then phase 3 being registrational. And oftentimes, because of the size of the populations and the complexity of executing on those trials, the time from first in human to a registration and approval was often more than 10 years. We migrated from there into targeted therapies, where again, dose escalation and design was still appropriate, but again, you sub-selected a population, a smaller population based upon the biomarker of interest linked to the targeted therapy. In many of these cases, and especially recently, phase 2 becomes the registrational cohort with the commitment, because of accelerated approval, a breakthrough designation, with a commitment to replicate those studies in a larger phase 3 trial. In doing so, we've been able to shrink the timeline from the early phases to registration and approval to around 5 years, and in some cases even less than that. Now, if we look at immunotherapy, we have many examples where we leverage an adaptive trial design where the dosing strategy led to rapid expansion into cohorts. And we even had examples where those expansion cohorts from phase 1 became the registrational trial. And there was a commitment to do post-market approval or post-market assessment to confirm those findings. And that's driven down the timeline from early in human to approval, in many cases, to under 5 years. So it's an extremely important consideration for how we think about trial design and execution, how we become more effective and more efficient in this process. Again, also in the oncology space, drug development, instead of being linear as it was in that first example of chemotherapy or cytotoxic therapy, it's becoming more iterative. We're learning as we go, so we learn and adapt to what those findings are, from discovery to the early toxicology studies to first in human to the registrational aspects of trials. And those learnings improve the ability to transition through these phases and allows for a faster and, in many cases, a more successful application across these various stages. For oncology, we're finding a variety of things that are helping to facilitate that. We are looking at and approaching different ways to look at preclinical assessments, better preclinical models, complementary preclinical models, patient-derived xenografts. We're looking at organoids, organs on a chip as a way to complement those traditional toxicology studies and mechanism of action studies that were often done in animals and non-human primates. Because of our better understanding of biology, we have biomarker assays that are being effectively used for patient selection and enrollment, also for monitoring and adverse events. And these include a whole spectrum of technologies from genomic to pathology to flow cytometry, and as appropriate, we see them evolve into companion diagnostics that are relevant for the safe and efficacious use of a targeted therapy. We're seeing those again applied for identification of the appropriate patient populations. And as we look at rare alterations that might occur in 1 to 2% or less of a target patient population, we're also leveraging what we know in the current world around patients based upon diagnostic testing to point patients to trials to increase that enrollment. And we've already talked about the appropriate and adaptive trial designs, whether they be, again, basket or umbrella trials, driven by our understanding of pathology. So the point here, the point of the last two slides are the way in which we approach trials, the way in which we approach molecule development, trial execution is becoming a much different arena. It's more flexible. It is leveraging our learnings to become better at how we plan for and execute on the various phases of development. We look at the drug and diagnostic co-development process, and this is most appropriately mirrored by the development of companion diagnostics, where you have parallel development of the novel therapy as well as the biomarker-based assay that's important as a differentiator and important to identify patients for the receipt of that therapy. Over the years and through my experience, there's a variety of factors that really are critical for success. First and foremost is early engagement. You know, we see now, as compared to what happened historically, the discussion with pharma, with diagnostic partners, with service providers, very early in the process, so that you can select the right assays, the right platforms, and it really becomes an integral part of the drug development strategy. We see alignment of parties to all of the stakeholders. So in pharma now, we have many, in biotech, we have many companies that have their own diagnostic experts. They understand the development of diagnostics. They talk the same language as in vitro diagnostic companies and service providers. So we understand better and more holistically the needs for that co-development, including timing. And those activities are often managed through a joint governance structure. And then lastly, I would say one of the things that's been relevant is flexibility and dialogue that flexibility comes from with regulatory agencies. Being able to ensure that a test is developed appropriately, that it's incorporated appropriately to determine both analytical and clinical validity, and that it meets all of the standards that the regulatory agencies that the FDA may have and apply, so that these novel therapies and the associated diagnostics can get to patients and really drive better care for those patients in a timely fashion. So early engagement, alignment of all parties, and dialogue and understanding how we can best do this with all parties, including the regulatory agencies. This next slide is just a snapshot of the historical approvals around companion diagnostics. I've been lucky enough to have been around long enough to be involved with those early development activities, including HER2 and the first targeted therapy in breast cancer, all the way through some of the recent developments. And what we see here is a really encouraging story in oncology, where we've seen this march, leveraging what we know about biology, leveraging what we know about diagnostic testings, and that convergence of diagnostics and drug development to continue to deliver important new applications to better care for cancer patients. So it's a great story, and as I mentioned, is expanding now to other therapeutic areas like immunology, rare disease, neurodegenerative disease. So the lessons that we've learned in oncology will be directly applicable to these other areas. I'm going to switch now and talk a little bit about applications and how this area has evolved and will continue to evolve, both around therapeutic options, but also around technologies that give us a better assessment of disease biology, better assessment of how patients might respond to a novel agent. The first one of these areas will be in the immunotherapy or immuno-oncology area. Great success by the checkpoint inhibitors across multiple indications in oncology. We see also the evolution of biomarker approaches with PD-L1, mismatch repair, tumor mutation burden, combination of biomarkers all being applied to better discern which patients will respond appropriately. We have other immune therapeutic agents that are in development. These are often activators of the immune system, so OX40 and others being examples of that. We've had great success and continued evaluation of adoptive cell therapy, CAR T therapies, TCR-based therapies, and I'll come and talk a little bit about that at the end. And then cancer vaccines, where we can leverage what we've learned from neoepitope production or development in tumors and how that engages the immune system. So it's a great area and an expanding area where each of these immune therapy applications will continue to evolve, and we'll see, and we are seeing where combinations of these could be of great value. So we have examples of companion diagnostics in this space. The first example is PD-L1 expression determined by immunohistochemistry. This is figures from a publication looking at patient response and stratification using a PD-L1 assay linked with the therapeutic Keytruda, with a 50% cutoff here, where 50% or greater of the tumor cells express PD-L1. And on the Kaplan-Meier curve on the right-hand side, the blue line, the better survivorship, are those patients with a high level of overexpression, and the gold line would be those with a lower level. So PD-L1 being a biomarker that's often leveraged today in multiple indications, either as a companion diagnostic, a complementary diagnostic for the development of checkpoint inhibitors across multiple indications. A second advance in recent years was the tumor agnostic approach for a companion diagnostic. This, again, the biomarker drives the decision-making around who would be eligible for a therapy and who should not be considered for that therapy. Here's an example of mismatch repair deficiency or microsatellite instability high being leveraged across multiple tumor types for consideration with a checkpoint inhibitor. And again, this is a quotation from the director at the FDA. Until now, the FDA has approved cancer treatments based on where in the body the cancer started, for example, lung or breast. We now have approved drug based upon the tumor's biomarker without regard to the tumor's original location. So really a great approach. We've seen other examples like this as well. Tumor mutation burden, a lot of interest in this space, again, leveraging the increased tumor mutation burden with a specified cutoff to identify patients who should be considered for an immune checkpoint inhibitor. This technology, there's a number of ways to measure tumor mutation burden. One of the challenges has been standardization around that, but just like with PD-L1 expression and the Blueprint trial, Friends of Cancer Research and laboratories and clinicians and across the industry have been looking how we best harmonize these different measures. Tumor mutation burden often being potentially a surrogate for the production of novel antigens, neoepitopes that would drive an appropriate response of an immuno-oncology agent or an immunotherapy for those patients with a high tumor mutation burden. Multiplex immunohistochemistry, we see more interest in this space. I think today mainly in the discovery area, where we're looking at biomarkers that reflect biology of both the tumor and the tumor microenvironment, PD-L1 on tumor or infiltrating lymphocytes, the type of infiltrating lymphocytes that are found in the tumors, and other molecules that would be representative of that interaction between the immune system in the tumor microenvironment and the tumor. And in other ways, we're looking at how to assess the complexity of those assessments by using machine learning and artificial intelligence to understand the level of expression of the biomarkers as well as their spatial relationship to one another. So again, a critical element of looking at not only what biomarkers are expressed, but the context of that expression. In that sense, we have the use of a combination of biomarkers here, identifying tumors that are cold. They don't express the right profile in the tumor, or they've excluded the immune system cells, tumor infiltrating lymphocytes, over to the right-hand side where you have a tumor that not only overexpresses PD-L1, but it has an increased number of infiltrating lymphocytes as defined by biomarkers that not only identify the types of cells, but also whether or not they're effector or regulatory cells. So again, another way of incorporating a picture of what's going on in the tumor and the tumor microenvironment. And then folks have looked at, okay, so we can do these things individually. Is there greater resolving power if we apply these tools in combination? And this is a study that was fortunate to be part of, but also looking at things like, if we look at immunohistochemistry, CD8 positive T cells, whether they are not in the tumor area, whether they are infiltrated or they're excluded. Mutational burden, a high mutational burden or a low mutational burden. And then even factoring into that, tumor inflammation signature. So are we expressing those genes within the tumor and tumor microenvironment that would say that the immune system is activated and really addressing that interface between the tumor and the tumor microenvironment. And in the table on the right-hand side, what you see is that individual biomarkers have a certain predictive value or certain performance characteristics around sensitivity, specificity, positive and negative predictive value, accuracy. But if we look at the third row here, the best performance is that combination of biomarkers. In this case, the type of lymphocytes, the mutational burden, and the expression of genes that would be an indicator of the interface and interaction between the tumor and the tumor microenvironment. We'll talk a little bit now about some other technologies that are relevant. So we've been involved with a technology. This is a technology developed by NanoString called digital spatial profiling. On the cartoon on the left-hand side, it gives a description of the assay. It's a nice combination of both a pathology-based assay, because you do have morphological features that can be assessed, as shown in the lower right-hand side, where you can not only look at morphology, but you can assess expression of proteins or RNA molecules within those regions of interest. So you can look specifically at the tumor, you can look at the associated tumor microenvironment, you could look at normal tissue around that area and quantify in a digital fashion the expression levels of, again, either proteins or RNAs. The way in which that is done is leveraging both antibodies that are used to detect proteins, but then linking those with an oligonucleotide probe that can be cleaved and then counted like you would in a genomics-based assay in a system, the nanostring N counter. Likewise, you can do the same thing for measuring gene expression using a nucleic acid-based probe, again, cleaving off the appropriate oligo and then counting it in the appropriate instrumentation to get a digital readout of expression. So the novel features of this are you are able to combine pathology with the digital assessment of expression of either proteins or RNAs. The next two slides are part of a feasibility study that we did looking at some head and neck cancer samples. And we evaluated these in a variety of methods, including multiplex immunohistochemistry, including gene expression for a tumor inflammation signature, but then also looking at protein expression on the DSP platform, and then also RNA expression on the DSP platform. And what you can see here, we had tumors that were cold, that they did not express or overexpress certain biomarkers like PD-L1, and they did not have a preponderance of infiltrating lymphocytes. And then hot tumors that again expressed both PD-L1 and then had associated TILs within the tumor area and tumor microenvironment. And what you see here is you can see differences in distribution pattern between those tumors that are considered cold and those that would be considered hot. Hot being those most likely to respond to a checkpoint inhibitor. In a similar fashion, we presented this at AACR last year. You can measure RNA counts. So again, get a digital expression of different genes that are relevant both to the tumor biology, but also to the expression of genes within the infiltrating lymphocytes in the tumor microenvironment. And again, as shown here, the sort of predetermined cold versus hot tumors, you can see a quantitative difference in the level of expression. So again, being able to collect multiple parameters that could be relevant to understanding in a better way which patients might respond or which may not respond, but also being a discovery tool to winnow through that list of proteins and or genes that you might want to incorporate into an enrollment trial. So again, great discovery tool and fits in nicely with the ability to marry pathology, morphology with measurements within the tissue sample. So other technologies that really are starting to move very quickly into this space are liquid biopsies technologies. And so there's liquid biopsy is a terminology that means many different things. It can be measured in many different ways, but it's really related to the fact that when a tumor is vascularized, as tumor cells are shed or they die, the cells themselves or the DNA content or protein content is shed into the bloodstream. You can measure the level of circulating epithelial cells or circulating tumor cells, count them in the bloodstream, and that was one of the first applications for liquid biopsy. The count of CTCs is often has been demonstrated to be a prognostic factor, but it's also been shown that it might be a good surrogate endpoint for measuring response to a given therapy. We can also take those individual CTCs and look at protein expression and genomic information, either on a slide, or by extracting those cells off of a slide and then extracting the nucleic acid content and assessing it for mutations or gene expression. We see a lot of work in the circulating tumor DNA or cell-free DNA, and those technologies, either the single gene, like the first example of a ctDNA assay that was approved, or by the agents, by the FDA, associated as a companion diagnostic with a targeted therapy, or they can be an NGS-based assay where you look at multiple genes and you're able to look at mutations within the relevant genes. You're also able to assess copy number variations as well as translocation, so giving you genomic information. And then also there's interest in exosomes being the lipid bilayer of cells, and enclosed within that are either proteins or nucleic acids that can be assessed. So a variety of ways to look within the blood for biomarkers that are relevant for understanding disease biology and therapeutic approaches. This next slide is a cartoon representation of how you might utilize these technologies in the management of patients with lung cancer. What we've seen is mainly applications on the right-hand side of this panel in advanced disease, where biomarkers measured, genomic biomarkers measured in the blood, because they correlate with genomic alterations found in the tissue, can be used for assessment and selection of appropriate therapy. We see applications for monitoring the level of that variant in the blood on treatment to indicate as an indicator of response or relapse. We can measure also whether or not the therapy is effective at a level that it's driving down the disease biomarker to a level that is low or undetectable, measurement of minimal residual disease. And if a patient has a relapse, we can look in the blood and look at resistance mutations. Likewise, we see interest on the left-hand side, where these panels may be used for early detection, that they could be used in cases like you've got a nodule seen on a CT scan in a lung cancer patient. Is that a cancerous nodule, or is it due to another reason? And then also you can use these for adjuvant treatment follow-up. So what we're seeing is post-surgery. So we're seeing a variety of ways in which this technology can be applied. And we've seen recent examples of and thinking around companion diagnostics using a liquid biopsy versus a tissue biopsy because of the correlation, the good correlation between tissue mutation spectrum and what's found in the blood, and the correlation of clinical responsiveness. Likewise, we want to use technologies that allow us to assess the complexity of disease biology. We know that tumors are heterogeneous. We know that when we look at expression patterns or genomic patterns within a single tumor, a biopsy, across multiple biopsies. So we know there's inter-tumor heterogeneity, intra-tumor heterogeneity, and intracellular heterogeneity. And that heterogeneity is expressed both spatially and temporally within the patient at a given time or over time as well. So technologies are being developed and being applied that will allow us to look at single cells to assess the level of that heterogeneity and how that's relevant for therapeutic response to a given therapy or therapeutic regimen. And here's an example looking in a blood-based cancer where longitudinally monitoring this patient over time on treatment 1, treatment 2, and during relapse, you can measure the evolution, the clonal evolution of that patient's cancer over time, starting with a given mutational spectrum. And as treatment duration is accomplished, and as we look at changing treatment, we can monitor these patients for that heterogeneity and evolution related to the therapeutic approaches. So again, a great variety of tools, whether they be multiplex, whether they be combination of technologies, whether they be profiling what is happening within the tumor, using blood-based surrogates for what's in the tissue, and monitoring at the single cell level how that heterogeneity translates into features that are relevant to response for a patient to a given treatment regimen. So very exciting time as all of these technologies converge and help us both in the drug development process, but also potentially in patient management. I'm going to move on now and talk about adoptive cell therapy and how biomarkers are relevant there. So adoptive cell therapy, again, it's a increasing area of interest. Great success with products on the market addressing, B-cell malignancies. But again, much like immunotherapies, each patient may have a unique profile. So we have to look at biomarkers that are relevant, genotypic, phenotypic, that reflect both the patient's cancer, the nature of the engineered therapy, the adoptive cells, the CAR T molecules, as well as the response of that patient. And so, that's challenging for us. And we have to also balance how these biomarkers are used to assess toxicity. So cytokine release syndrome can be very relevant for patients with receiving adoptive cell therapy, biomarkers of efficacy, and relapse. How all of these come together is a very interesting story. So again, it's critical for patient identification. Are they expressing the target antigen of interest? For looking at pharmacokinetics and pharmacodynamics around the treatment dose, the persistence, and expansion of the cell therapy when it's administered to the patient. Does it engage the right target? Is the target population decreasing? Can we measure it? Is there minimal residual disease, or have we eliminated the clone? So technologies that are relevant and biomarkers that are relevant in this space are the assessment of the CAR T molecules, for example, in a CAR T project or a CAR T therapy, where we're looking at both persistence and expansion of the CAR T molecules, CAR T cells. Characterization of the response to the CAR T, immunophenotyping. Are we activating the appropriate immune response? The PK-PD relationship, so the engineered cells being the therapy, we're looking at those as well as the tumor cells that the CAR T therapy is targeted against to assess the level of the CAR T molecule as well as the response of the tumor cells. Are they decreasing? Are they increasing, et cetera? Mechanistic markers of efficacy. So if we measure the target antigen, CD19 positive B cells, are they decreasing as measured by flow cytometry or by molecular diagnostics? Patient ID and stratification. If we're looking at CD19 positive tumors or BCMA positive tumors, can we select and identify the right patients? And in the middle, second row, predictive biomarkers of safety, response, resistance, or relapse. So minimal residual disease, but also looking at the immune response to the adoptive cell therapy. Is it a heightened immune response? So you want to activate the immune system, but you don't want to overactivate it so that you cause challenges with that overall immune response, expanding beyond the target cancer cells of interest. So what's been suggested in adoptive cell therapy is we take a systems type approach. You have to measure multiple parameters, both of the therapy, the engineered cells, of the tumor population, of the response, the immune response of the patient. And those tools and technologies can be very broad. They can be genomic technologies, they can be proteomic, cell-based technologies, etc. And so again, using those types of tools and applying them to the challenges and opportunities with adoptive cell therapy, looking at, again, that interaction of the engineered therapy, the target population, and the patient's immune response requires, again, a different thinking than we've applied in more traditional oncology therapies. Now, for each of these, whether it's a biomarker assay, one of those 300 or more that are in current drug labels that look at safety, efficacy, toxicity, etc., whether it be a companion diagnostic that's necessary for the safe and efficacious use of a therapy, we have a variety of ways that we can commercialize these assays. Many biomarker assays are lab-developed tests. There are assays that give us information around the patient and the patient's response to a therapy, but they are developed and offered by laboratories with the appropriate accreditation. There are examples where an in vitro diagnostic company, a lab service provider, and a pharmaceutical or biotech company have worked together in a three-way partnership, linking a kit-based assay that can be broadly distributed post-approval of the drug and diagnostic to link that development so that on day one, you have the approval and a model that allows access to testing and access to therapy on day one. We also have examples where, again, showing flexibility of the regulatory process of a single site offering the test. They take it through for regulatory approval just like a distributed kit, but it falls all within the quality system of that single location. And we've got examples of those as well. Those often work very well for novel technologies or areas in which the overall patient population might be somewhat limited. And then on the right-hand side of this slide, when you think of commercialization, and this is something that pharma and biotech, in vitro diagnostic companies, labs have really started to collaborate very effectively on. On day one, when the test and therapy are available, there's a whole variety of things, a very long checklist that must be worked through in order to ensure success. So is the lab up and operational? Do they have the right equipment? Is the test on the test request form so that clinicians can order it? Are the appropriate lab staff, including pathologists, trained on the complexities or the nuances of the assay? Are reports ready to go out? Can we take samples from their collection and from the patient, liquid biopsy, tissue sample, move them to the laboratory appropriately so that data can be generated and the test results can go back in a timely fashion so patients can receive the novel therapeutics that they are in need of? Can we educate and communicate appropriately so that all parties understand how the test works and what value it provides? Are the client call points appropriate? So are we talking to pathologists? Are we talking to oncologists? Are we talking to healthcare providers and payers so that there's no weak link in the chain? And then again, is the right coding and reimbursement strategy established. So it's a very complex set of things that need to be addressed. And until very recently, it was often believed that, day one, you put the test on your test menu and everything is fine. But there are many, many, many things that have to be established so that an effective use of the therapeutic and diagnostic can happen. So my last slide. So what I tried to do today is talk a little bit about how biomarkers, we have to think about them from exploratory use, mechanism of action, all the way through clinical development into commercialization. We know that there's a variety of ways of thinking and technologies being applied to how we look at biomarkers, moving from single gene, single protein to a more broad spectrum multiplex technologies like NGS, like multiplexed immunohistochemistry. We know we have to consider the right development and commercialization strategy. Will it be a lab-developed test because of its intended use? Does it need to be a companion diagnostic? Do we need to have a single location? Do we need to be able to distribute that product more broadly? We talked about incorporation into the drug development process and how it's iterative. It's not linear like it once was. So biomarker-driven trial design and execution, application of biomarkers from early on in the drug development process through commercialization. The ability to expand these applications to other areas outside of oncology, that being driven by better understanding of disease biology, the relevant biomarkers, examples, rare disease with gene replacement-based therapies, CRISPR technologies, neurodegenerative disorders, and other immunologically based therapies. And lastly, I would say that if we look at the regulatory approaches, the flexibility to ensure that we meet all of the standards that are necessary for both the diagnostic and the drug development, but we look at how we can flexibly work together to advance these new therapeutics and the associated diagnostics in an appropriate and timely manner. And lastly, thinking of the global regulatory environment, that's an area that we still need to work on as we think about the development of these tests in different geographies, whether it be the US, North America, Europe, and Asia Pacific. So with that, I'll conclude. So thank you for your time today. It's been a pleasure being with you and presenting some of my thoughts of where we are today and I think where we're heading in the future. So thank you again.